Okay, so I want to talk to you briefly in 15 minutes about our research on the neurogenomic basis of adaptation to captivity. And obviously, we use the zebrafish as a model. And I can't really go into everything that we do. And so what I'm going to do is give an overview of some of the transcriptome work that we've done, and then try to highlight some mechanistic experiments we use to follow up one of those pathways, because I think it fits in very nicely to some of the other work that we presented later in the symposium. The motivation for our work is primarily to discover the genetic basis to adaptation to captivity, and we obviously focus on behavior. And here I've shown 3 different instances where we rear animals in captivity. This is salmon in a hatchery, which is primarily focused on mitigation of population declines. So it's a conservation-based captive rearing. This is dinner, chickens, and this is some sort of perverse death wish in a zoo down in South America where they actually let you ride lions. Don't try this at home. All 3 of these examples are cool because they demonstrate one of the, what I think is a very profound and cool observation about adaptation to captivity. And that is almost universally, whether you look at this trait in fish, birds, or mammals, you observe decline in fearfulness, or reduction in anxiety, or most lay people would call it tameness. So what does that look like in zebrafish? That seems like a strange transition to go from someone riding a lion to zebrafish. But what we primarily do in my lab is we examine differences between zebrafish captured in the wild and animals that we call domesticated, that your canonical lab strains. This is a strain which we call Nadia, which is a couple of generations removed from swimming around in the wild in India. I want you to pay attention to their place preference behaviors as well as their startle response. They, I would argue, don't like the technician waving at them. And they tend to hang out at the bottom of the tank and away from the stimulus, in this case, the finger test, which I have yet to publish. Now, what I'm going to show you is a lab strain of zebrafish now to show you the difference. Now, the difference here is a little bit subtle, so unlike, Alan was saying things can be black and white. Well, let's see if you can pick up on it here. So we're going to have the technician wave to the zebrafish. Now we'll do the finger test, which I've yet to publish, but and I think most of you who work on these animals that work on lab strains are probably more used to the finger nibbling phenotype than you are the freak out phenotype. All right. So because this is more of a biomedically oriented conference, I would argue to you that that really is a genetic difference in anxiety-related behaviors. And you can argue with me in question time whether or not you buy that. But if you're an evolutionary biologist, obviously we talk about the transition from a wild-type behavior to a domesticated-type behavior. But we're really interested in asking big questions about whether we can identify general patterns, such as what we observe in stickleback with parallel genetic changes resulting in pelvic spine reduction, or if we see a whole bunch of different genetic pathways to the same phenotypic endpoint during domestication. And generally, we take a research approach that is twofold. In the first approach, we look at things comparatively. We look at those wild strains and those lab strains. And we use various techniques to quantify those videos that I showed you there, which I learned the hard way in my postdoctoral time, that ethology is not easy. And then we use transcriptome analyses to identify candidate genes and pathways, and then undertake hypothesis testing of those candidate genes and pathways to try to figure out mechanistically whether those genes actually relate to the behavioral phenotypes. The work that I'm not going to talk about because of time is our selection experiments, where we're creating inbred lines of anxious and bold animals that recapitulate those phenotypes I showed you. And in that case, it's a more quantitative genetic approach where we're estimating things like heritability and genetic correlations, looking at genetic constraints on evolution. But both approaches, ultimately, we're applying SNPs and RNA-seq to identify polymorphisms that we can then associate with the phenotypes themselves using QTL and genome-wide association studies. We We've done a lot of different transcriptome analyses. Obviously, because we're looking at behavior, we decided to look at the brain first. The central hypothesis that defines most of these studies is this, that the differences in behavior that we observe among wild, or to you guys, anxious and domesticated, or whole strains, can be explained by patterns of gene expression in the brain. Now, the general approach is obviously to take those behaviorally distinct strains of animals and we replicate wild and domesticated strains so that we're not using essentially one wild and one domesticated in comparison. We take those behaviorally distinct animals and we dissect out their brains, and when we first started, we did whole brain transcriptome analyses, either with pooled brains or on individual brains. Of course, the reviewers, especially if they're mouse people, they don't like that. And so we've moved now to dissections of brain regions and amplification of the RNA to look at region-specific changes in the transcriptome. And we submit that or interrogate the transcriptome with a variety of tools. Started with Affymetrix arrays and moved to Nimble arrays, and now obviously the more recently developed tools like RNA-seq using 454. Now, we've done a lot of these different experiments, and I want to show you some of the kinds of results that emerge and the approach that we take. We try to replicate our experiments using different techniques, because microarrays, as you all probably know, tend to give you a lot of false positives. A lot of times the data look like this, where the columns here are individual microarrays clustered by their behavioral type, either the wild anxious type or the lab friendly type. And these are 251 genes that came out as differentially expressed in the brain of the zebrafish. And what this is essentially showing is that there's a group of genes here that are downregulated in anxious animals relative to bull animals and things like that. So, as you know, if any of you have done genomic work, you do a transcriptome experiment like this, and what you get is a massive list of genes that are different. And you don't know what any of them do. None of them. Right? Because you're an evolutionary biologist, you're not a cellular biologist. And so you bother your collaborators extensively to try to figure out what they do. Now, genomic experiments are expensive, and so we try to follow up on these experiments with more focused approaches where we take genes and validate them on independent samples that were not associated with the microarrays, to ask whether the genes that we've identified that are differentially expressed between anxious and bull animals are actually real. And you can see, so what we have here are the microarray results. For example, this is a gene I'm going to talk to you about in more detail that shows that anxious fish generally have a downregulation of selenoprotein V relative to the bullfish. And then when you do a new 100 animals and ask, is that really true? And you see a recapitulation of that pattern. The other approach that we take is to then take our genes that we've identified that a lot of times we haven't ever heard of and ask whether similar studies in other organisms, because we studied domestication in lots of other charismatic organisms like the famous fox domestication studies and dog-wolf comparisons, and their work in mice and rats on domestication. And we ask whether those kinds of genes have ever been identified in these kinds of studies as well. Generally, we do see, we've yet to see a gene that comes up in all of them, but we definitely see genes that come up and pathways that come up in more than one. One such that I'm going to focus on is a gene that I had never heard of, and we tried real hard to falsify it in my lab, is this selenoprotein P, which we observed, and I showed you the data for that in zebrafish, and also turned out to be differentially expressed in comparisons of dogs and wolves. Now, we take all of the genomic data and we essentially try to synthesize patterns and figure out, are there common themes here? Are there common pathways and sets of genes that appear to be effective or appear to be evolving in terms of expression patterns during domestication? We followed up on a whole bunch of these, the ones we've done kind of the most work on. How am I going for time? Okay? Time-wise? Some of the pathways that we've done the most follow-up work on are selenium homeostasis, which I'll speak about in a minute, as well as divergence in orexigenic neuropeptide signaling, the neuropeptides that make you feel full or hungry, We've done a ton of follow-up work, especially in the selection lines on the stress pathway, that have been a result of the transcriptome analyses from our comparative work. Now I chose to, I obviously don't have the time to go through all of those things, and I chose to focus on the selenium story because I felt like it fit into the literature about the symposium in terms of biological psychiatry and neurological phenotypes. This is the pattern we observed. We originally saw selenoprotein P coming up as differentially expressed in our experiments, and because we'd never heard of it, we ignored it for a really long time. But it kept coming up, and eventually we got to the point where we were like, well, now let's try to falsify it. And it kept coming up, and it kept showing us patterns like this. Here's one of our indices of behavior. We're only now transitioning to video tracking, so we do more, I'm going to say, crude analyses compared to the folks that Noldus and Hamil's lab and Noldus lab. This is essentially the proportion of time spent near the observer. And this is an anxious animal. They don't like you. This is a bold animal. They're in love with you. And you see a pretty strong correlation, especially for a gene expression and behavior, across 3 populations. Between selenoprotein P and observer orientation. This is a gene that transports selenium throughout your body and maintains selenium homeostasis in the brain in humans and mice and fish. And it turns out when we started really looking into this gene in terms of follow-up studies, selenium is very necessary in terms of a micronutrient for what we call normal brain function. And if you have selenium deficiency, that has been associated with a variety of pathologies in humans. And there's considerable interest in the role of selenium supplementation, these, among a whole bunch of other things. But in terms of mental health, for treatment of things like disorders of mood, like depression and anxiety. So let's look at the literature very briefly about what does selenium supplementation do in humans. So there are studies that show selenium supplementation in humans has a beneficial effect measured in a variety of different ways that the human folks use. On mood, including decreased anxiety and whatever elevated mood means. Anyway, these are a variety of published studies that show that selenium supplementation does have an effect. However, there are equally compelling studies that are very large and very well controlled that show no effect whatsoever. And recent work has also shown that the response measured by activity of certain enzymes in the blood plasma is actually a complex interaction between gender, body mass index, and genotype, particularly SNPs in the selenoprotein P gene itself. So, open questions in the human literature are actually, does selenium supplementation influence behavior? They don't know, conflicting data. And are the behavioral and transcriptional responses, like the physiological plasma response, dependent on sex, body mass index, and genotype? And I know Dr. Gerlach already knows the answer to the third one. So we did selenium manipulation experiments to follow up on this, to ask essentially, if we supplement these different strains of animals with selenium, can we change their transcription profiles of various selenoprotein genes in the brain? And can we change their behavior? It's perfect, almost done. And what you see here is, yes, we can change transcription profiles, but it really depends on the strain. These are 4 different strains. These are wild, these are domesticated. This is the only strain, the one with the lowest expression of selenoprotein P, that showed a significant response to selenium supplementation. We do use 2 different types of selenium. Similarly, if you take this strain that we would have said is a non-responding strain, and you do a new time course experiment, and you look at males and females, and you see this is the difference between supplemented and control animals. You see females upregulate selenoprotein P early in the experiment, and then don't, whereas males don't upregulate selenoprotein P expression in response to supplementation until later in the experiment. In terms of their behavior, we did observe a small but significant change in observer preference to selenium, where we saw supplemented animals did get more bold than anxious animals, or than control animals, sorry. But for many of our behavioral indices, again, we saw population-specific responses, where certain strains responded in that they changed their depth preference and actually preferred to swim deeper in the tank, and other strains actually elevated their position in the tank on selenium supplementation. So in terms of the actual contribution of the zebrafish model to human-related biomedical questions, I guess the summary here is that we use transcriptome-based studies to nominate candidate pathways, and then use the zebrafish model to answer the question of whether or not the behavioral response to selenium actually can depend on gene type, population, sex, things like that, and effect in humans. And so I should acknowledge that a lot of the selenium work was done by my graduate student, Maya Benner, and the funding primarily came from the National Science Foundation. And if I have time, we'll take questions, or we can wait.